Now let's talk a little bit more about antibodies, and then we're going to also talk about the systemic response of the body to an infection or inflammation. So what an antibody is a Y-shaped molecule. It is a very complex protein, and we'll talk about the structure in a minute, that attaches to an invader. And you can see here that this invader, this pathogen, is now red flagged. So you can see it calling on its friends, viruses and other bacteria, but they are telling them, get away from us because you've been marked, and the immune system is now going to be able to identify you. This is what an immunoglobulin looks like. It is made out of 4 polypeptide chains. It is arranged in the shape of a Y. 2 of them are long or heavy chains, while 2 short or light chains. The stem of the Y is known as the constant end, and all of our immunoglobulins have these exact same structure when it comes to the constant end, but they do differ in here, known as the specific antigen binding sites. And because we have so many different antigens, these obviously are going to vary. Antibodies are responsible for the humoral immunity because these antibodies circulate within the plasma. And there are 5 different types of antibodies. They are known as gamed. So IgG, A, M, E, and D. And in order to know, you want to be able to answer all of these questions when it comes to these antibodies. So which ones are able to activate complement? And we should know that by now, that those are the IgGs and IgMs. Which one is the most abundant? Well, IgG is number 1, followed by IgM. Oh, in order to remember which ones that activate complement, those are GM. Okay, G and M, General Motors. Now, which one is found in exocrine secretions like milk? So our exocrine secretions like milk and sweat and so on have their own antibody, and that is IgA. Now, which one attacks parasites? Would be involved in allergies. That is IgE. So allergy, IgE. Remember when we said if you get an incompatible blood transfusion, that would lead to agglutination. You want to know which one of these antibodies would be responsible for the agglutination process, and that is IgM. Now, some of these antibodies have the ability to cross the placenta and go from mom to baby to provide some immunity, and that is IgG. Now, what do antibodies do? Okay, they act as an opsonin, kind of like that little comic that we saw. They red flag the pathogens, and that leads to the enhancement of phagocytosis. IgG and M have the ability to activate the complement system. They provide what is known as the antibody-dependent cellular cytotoxicity, so basically killing cells, but depending on the presence of antibodies. And they sometimes can directly neutralize toxins and viruses. So we do have antibodies that are antitoxins and have antibodies that are against viruses, like the antiviral proteins that we've discussed previously. And here you can see its function as an opsonin, where that Y-shaped antibody attached to the bacteria, red flagging it, the phagocytic cell is able to identify or has receptors for the constant portion of the antibody. We're able to identify them. Now that phagocytic cell will engulf that bacteria. Remember we said that IgG and IgMs activate complement, and that's because they have these little binding sites for C1. Remember, C1 was the first of the complement system. In order, we would have to activate that first protein. That was the classical activation pathway. Antibody production really depends on if the immune system if that's the first time to see the pathogen, or if we already recognize the pathogen by our memory cells. So if it was the first time that the body is introduced to that antigen, it's going to take time for the body to do all of the process that we talked about, the antigen presenting, the activation of the different cells, and then the making of the antibodies. So that first response, known as the primary immune response, takes a little bit of a time. It's a slower process. But at the end of that process, we make memory B cells and memory T cells. Those remain for longer periods of time, sometimes even lifelong. So that if that same pathogen comes in again, the memory T cells recognize them right away, and that leads to a very fast response, and that is known as the secondary immune response. And this is the whole idea of vaccines. Vaccines, we obviously don't give the actual antigen, we give little parts of the antigen, or sometimes we kill the antigen, or kill the pathogen and give that. So that when you are exposed for the first time to the vaccine, your body makes memory cells, memory B and memory T cells, so that if your body is exposed to that antigen again 10, 15, 20 years later, it still has those memory cells and it could identify them right away and again lead to the fast production of antibodies, that process called secondary immune response. And here you can see the difference where that primary immune response, the very first time the body is exposed to an antigen, it takes about, these right here shows the time in months. So it takes about 2 to 3 weeks in order to produce a maximal response. But again, we have memory cells. These memory cells remain with us. Some can remain for 10 years. Some even can remain lifelong. And that's why sometimes some injections do have to be repeated because we lose those memory cells, while others we only have to take them once or twice. Again, if we're exposed a second time to that antigen, our memory cells recognize them right away, and you see pretty much an instantaneous response and making of antibodies against it. There's two kinds of immunity that we would like to introduce. There's an active and passive immunity. Active immunity, that is the permanent kind of immunity, where the body responds to an antigen and produces its own antibodies. So the body is really actively involved in making the antibodies. For example, if we get sick, we make antibodies against that pathogen. And if the memory cells remain for a long time, we become immune to that disease. Sometimes memory cells, though, do not last for a long time. So we might be prone to getting secondary, infected again and again with the same antigen, depending on how long the memory cells remain in our system. Again, that is the same concept of vaccines. Again, vaccines, we were not going to give the actual living antigen. We could give small parts of it, so very tiny antigenic parts of it, or the dead pathogen. Sometimes we can give small quantities of toxins or very harmless antigenic molecules, basically the parts that the immune system needs to identify what that pathogen was. The second kind of immunity is known as passive immunity. This is a temporary kind of immunity where you are handing over pre-made antibodies to the immune system. And it's named passive because really the immune system didn't do anything. All it did was received those pre-made antibodies. Sometimes the antibodies, for example, transfer from mom to baby, to the fetus, and those would be the IgGs that can cross through from the placenta, or IgAs, and that would go again from mom to baby through breast milk. So these are important antibodies that are going to transfer passively to the baby, so provide the baby with some passive immunity for maybe a couple of months, maximum 6 months, 9 months maybe, and that gives the chance for the baby's immune system to develop and now perform, able to make their own antibodies. Now we're going to talk about the systemic response to the infection, known as the acute phase response. So with all of this that's going on, all of these different cytokines, there are different things happening in the body. So we feel sick, we feel fatigued, there's loss of appetite, there's a fever. Now, how do all of that develop? Well, there are different cytokines. So there's interleukin-1, for example, tumor necrosis factor, interleukin-6, that are being made by your monocytes and macrophages. They go through the bloodstream to the different parts of the body. So you see here that when it reaches the brain, it is going to induce a fever. It decreases our appetite. It is definitely going to make us fatigued and tired and sleepy. When these cytokines reach the liver is going to do something very weird. It is going to store iron and zinc. And a lot of the microbes need iron and zinc to survive. So we're basically starving the microbes from their needed iron and zinc and basically killing them that way. These cytokines will also reach the bone marrow, stimulating it to make more leukocytes. When these cytokines reach the adipose tissue and the muscle, that is going to lead to the breakdown of these compounds into, free fatty acids and amino acids. Remember again that we're really not eating, so we do have to find a source of glucose, and we could change these free fatty acids and these amino acids in the liver. Those can be changed into glucose through gluconeogenesis, and the body can use that for energy. These cytokines are also going to reach the hypothalamus and the anterior pituitary, stimulating CRH and ACTH secretion, which will stimulate the adrenal cortex to make cortisol. And this is going to end this part. The last part is going to talk about the abnormalities of the immune system.